Candida albicans. How does the infection start? Candida albicans is part of the natural microbiota in most healthy people's bodies. The infection starts when the fungus becomes opportunistic. This occurs for different reasons. Primarily, it occurs because the host is immunocompromised or because there is an imbalance of competing microflora. The parts of the body that could become infected with Candida albicans are the human gut, more specifically the GI tract, genitourinary tract, and skin. In rare cases, it can infect the bloodstream, and that is usually fatal.
Epithelial cells, which are part of the innate system, are our first line of defense in defeating Candida albicans. These cells include phagocytes such as polymorphonuclear neutrophils, macrophages, and dendritic cells. The polysaccharide structure of the cell wall of Candida are recognized by two extracellular pattern recognition receptors, Toll-like receptors and C-type lectin receptors, that are expressed by the innate cells. Once PAMs located on the pathogen are recognized by these receptors, a signaling pathway takes place where the chemokine and cytokine production and phagocytosis of Candida takes place. Once Candida is phagocytosed, its PAMs further activate a specific intracellular Toll-like receptor 9, which is located in the lysosome and is able to recognize Candida's DNA, or Knot-like receptor pyrin 3 within the inflammasome activation that activates cytokines interleukin-1 family. The TNF-alpha, which is a cell signaling protein, combined serves a protective effect against the pathogen.
What signals do they send to the rest of the immune system and how? When Candida albicans enters the body, its PAMPs on its fungal cell wall are recognized by PRRs on the DC cell surfaces. The DC cells then phagocytose and degrade the yeast cells, which provide a source of exogenous protein that is processed into antigenetic peptides. Then, the fungal peptide antigens are assembled onto class II molecules of MHC2, followed by the presentation of antigen to naive T cells. The T cells sample the antigen using TCRs. If the antigen is successfully recognized, specific cytokines will be secreted, which will drive the activation of one of three helper T cell subsets. Route 1 could be the secretion of polarizing cytokine IL-12, which then would induce master gene regulator T-BET, which regulates effector cytokine IFN-gamma, produced by the Th1 subset. This helper T cell subset is responsible for clearing intracellular Candida albicans infections and activates macrophages. Route 2 could be the secretion of polarizing cytokine IL-6, which then would induce master gene regulator ROR-gamma T, which regulates effector cytokines IL-17A, IL-17F, and IL-22, produced by Th17 subset. This helper T subset is responsible for clearing extracellular Candida albicans infections in mucosal sites of the body. Route 3 could be the secretion of polarizing cytokine IL-4, which then would induce master gene regulator GATA3, which regulates effector cytokine IL-2 produced by Th2 subset. If this occurs, it can promote growth and dissemination of Candida albicans.
When dendritic cells or DCs ingest Candida and is internalized, this leads to a processing and presentation of Candida-specific antigens via MHC class II. They are able to distinguish between yeast and hypophase form and induce T helper cell differentiation, and so this bridges the innate and adaptive antifungal response. Symptoms differ depending on the type of infection and where the infection is taking place. The most common symptoms include irritation, itchiness, UTIs, redness, swelling of these creamy white lesions on the tongue with a cottage cheese-like appearance as seen on this picture on the left, which is caused by oral thrush. This is an infection that occurs when the yeast infection caused by Candida develops on the inside of the mouth and tongue.
The way the immune system clears a Candida albicans infection is by the lectin pathway of the complement system. This pathway is initiated by mannose-binding lectin binding to Candida albicans surface carbohydrates. The end result of the complement system If it is successful, would be to either recruit inflammatory cells to help combat the infection, opsonize Candida albicans, making it more desirable to be phagocytosed, or induce the MAC attack, killing Candida albicans. All routes lead to the resolution of the infection.
Now, depending on the type of infection caused by Candida, the most common treatment includes antifungal drugs. So for example, for a Candida UTI, fluconazole is a popular antifungal that is used to treat the infection. For genital yeast infections, treatment includes over-the-counter antifungal creams or pill suppositories. For oral thrush, antifungal drugs nystatin or clotrimazole or an oral form of fluconazole are used. And for mucocutaneous candidiasis, which is a skin infection, antifungal cream and steroids are part of the treatment.
Candida albicans is a eukaryotic organism. Many of the membrane structures, proteins, and surface receptors it uses to invade host tissues have similar properties to human cells. For this reason, candidiasis and other fungal infections can be hard to treat. Developing new antifungal treatments that directly target the pathogen or support immune responses against it without targeting our self cells as well can be challenging. This is why candidiasis is so pervasive among individuals with immunocompromising conditions such as HIV, diabetes, cancer, and autoimmunity disorders. In healthcare settings, Candida albicans is the leading pathogen responsible for candidiasis. Public health officials, like the Center for Disease Control, have provided recommendations to prevent transmission of candidiasis and protect immunocompromised patients.
What are the important open questions that have yet to be investigated, and what are our hypotheses about this? We think that one of the bigger questions when talking about candidiasis is how can we prevent infection, especially in those patients who are receiving medications that make them more susceptible to candidiasis and are receiving health or hospital care. Considering Candida species are in our microflora, studies have shown that consumption of broad-spectrum antibiotics eventually leads to overgrowth of Candida. This is especially true if the patient that is receiving antibiotics is not concurrently receiving antifungal medication. However, as many studies have shown throughout the years of medical history, overuse of antibiotics lead to an increase in resistance by bacteria. Consequently, the same has been observed for overuse of antifungal drugs and an increase in resistance by fungi. We've conjectured the possibility of implementing new protocols to administer probiotics in an effort to keep the balance of all microflora while a patient is on an antibiotic schedule, and in the hopes of not increasing resistance of fungi and bacteria. Additionally, the infection of a patient by an exogenous species of Candida is not uncommon and is most prevalent in patients with intravenous catheters, as the Candida tends to colonize on the skin in this location. Outbreaks often happen between patients that have been infected by the same isolate, and it has been suspected that manipulation of medical devices by healthcare workers play a key role in transmission. It has been noted that healthcare workers with artificial nails are a possible source of isolates that cause these outbreaks. Our proposal would be to implement a protocol in which healthcare workers will no longer be allowed to wear artificial nails in an effort to reduce the risk of infection from patient to patient, and to enforce proper hand washing and IV care techniques. What is the social, global, and economic impact of this infection or disease? Currently, C. albicans is one of 5 species accounting for 92% of cases of candidemia, and this species alone accounts for 62% of all cases, so it is the most prevalent. In 2014, this fungal infection had an attributable mortality of 15 to 35% in adults, and 10 to 15% in neonates when in a hospital setting. Not only is this type of infection on the rise, it is also costly and has been reported to cost $40,000 per episode. Most notably, the annual number of causes of sepsis caused by fungal organisms in the United States alone has increased by 207% between 1979 and 2000. Additionally, C. albicans remained the most common species causing invasive candidiasis worldwide, although it has been observed that geographic location affects the prevalence of candidiasis by C. albicans. For example, occurrence has been as low as 37% in Latin America, yet as high as 70% in Norway. Identify at least one laboratory technique we reviewed this semester that could be used in this investigation. Research has shown that the quicker candidiasis is diagnosed, the lower the mortality. Tests that can aid in diagnosis of Candida would be the conduction of an ELISA to detect proteins.
The high prevalence of candidiasis has sparked interest in learning more about C. albicans and other related fungal infections for many years. Here at Cal Poly Pomona, students and faculty have undertaken research on Candida albicans.
So hi, my name is Benjamin Soto. I am a fourth year undergraduate in biotechnology here at Cal Poly Pomona, and I work in Dr. Nancy Buckley's lab in the biological sciences department. So my work focuses on really investigating how garlic is going to modify macrophage activities in response to several different types of pathogenic challenges. One of the ones we use is Candida albicans. So, Candida albicans is heavily prevalent, particularly in hospital-borne infections, and for immunocompromised individuals, it poses a real problem. So what we're trying to do is see when macrophages are challenged with, we use a heat-killed model of Candida albicans, how is garlic modifying the macrophage response primarily at the cytokine level? So we look at mRNA and we look at protein. So pathogens like Candida, they tend to be very dangerous for immunocompromised individuals, and I think a lot of people are interested in moving away from usage of pharmaceutical agents. So kind of what we're looking at are more natural remedies, you could say, that may be beneficial to individuals who have been infected by a pathogen such as Candida albicans. So I think finding these types of agents that could be utilized to help the immunity of these individuals who are sick is really beneficial to allow people to kind of broaden their horizon and say, well, what other types of agents are having the same effects, or what can we design synthetically that is going to mimic the effects and reduce the side effects that other medications may give, but amplify the effect that we're looking for. Okay. So my work primarily focuses on macrophages, and so usually I'm doing a lot of cell culture, doing a lot of in vitro challenging. I do some mouse work where we inject mice with a chemical called thioglycolate, and that kind of calls all of the macrophages to the peritoneum, and then we can collect those macrophages and then challenge them once we get back up to the lab with those cells. And so we treat them with the garlic. Then I collect their supernames and we perform ELISA analysis to analyze TNF-alpha secretion, and I also perform qPCR, so that's RNA extraction and isolation, cDNA synthesis, and the quantitative PCR to quantify changes in expression at the mRNA level of TNF-alpha, IL-6, and IL-1 beta, which you probably know is some of those downstream cytokines from NF-kappa B signaling.
Faculty at Cal Poly Pomona have also made breakthrough discoveries in research on fungal infections like candidiasis. Dr. Jill Adler-Moore made a breakthrough with the development of Ambizome, an antifungal drug used to treat infections like candidiasis in patients who do not respond to traditional antifungal drugs. This drug provided new tools for healthcare professionals to continue fighting the fast-growing, spread, and growing antibiotic resistance of fungal infections. With a high prevalence of candidiasis, there is no surprise that pharmaceutical companies, healthcare institutions, and academic institutions continue to fund research on Candida albicans. Pharmaceutical companies have an investment in this research that can return big profits if new drugs are discovered to treat candidiasis and related fungal infections. Immunocompromised individuals, hospital patients, and the general public would also benefit from continued research on Candida albicans, as physicians and healthcare providers would have new tools to help them combat persistent fungal infections that threaten them. Candidiasis is just one of the hundreds of types of infections that immunocompromised individuals, hospital patients, and everyday persons experience as a result of microbial pathogens. As research on the organism continues, the scientific and medical community are expanding their understanding of Candida albicans and the immune response it elicits upon infection of our tissues. With this information, it is hoped that immunocompromised individuals are provided an opportunity to fight fungal infections, improve their conditions, and continue to live fulfilling lives. Thanks for taking the time to watch our video.